in France in the Grande École. Then she had the PhD at the University of Liverpool, and then she joined the drug delivery section of the pharmaceutical science department in Padua. And she will present a work entitled Synthesis and Characterization of a Bone-Targeting Anti-Tumor Drug Polymer Conjugate containing a cathepsin K sensitive spacer.
Good evening. I'd like first to thank the committee for giving me this opportunity to present. intermediate molecule which is protected, and then the last molecule which is deprotected. So we did it, and we obtained all the steps were characterized by mass spectrometry. Then, so we have the alendronate prodrug, the paclitaxel prodrug. So now we have to look after the polymer carrier. So as I said before, there are neighboring alcohol in pullulan. So this can be used to form aldate functions by periodate oxidation. So it's what we did, and we obtained a 20% molar oxidation of paclitaxel, of pullulan. Then we had all the pieces of the puzzle, and we added them together by first adding the paclitaxel prodrug, reducing the Schiff bases formed using sodium acetylciborohydride. And then we added the pegylated alendronate, and we reduced all the bases, the Schiff bases, and also the aldehyde functions, which were unreacted because it can be toxic. And then we found in water only a part was soluble in water, the other part precipitated. So we characterized the soluble part by sec analysis, and we found that although the oxidized pullulan of initial oxidized pullulan was 40,000 molecular weight, the soluble part of the modified pullulan was 80,000, so it seems that only high molecular weight pullulan is able to bear the hydrophobicity maybe of paclitaxel, and that's why only this part is soluble and the other part precipitates because there is no equilibrium between hydrophilic. And then the dynamic light scattering showed we don't know if it's, and we don't yet if it's self-assembly, or we need to define a little bit more and do more tests, but it would be entity of 400 nanometers, and so these are quite fresh results. So so I'll conclude by saying that we made the prodrugs we want, we made the final polymer we wanted, and we have to work maybe on getting something a bit more soluble in water and characterize, of course, better the assembly we might have. And once this is done, we would do some biological tests. So I'd like to thank my group of Professor Paolo Caricetti, and also the groups of protein chemistry where I did peptide synthesis. And then I thank you. This is a photo of Padua.
Thank you, Dr. Bonzi, for the very interesting presentation. The paper is now open for discussion. Any question? Yes.
Your macromolecular prodrug is the definition has 3% of paclitaxel in weight. Did you have an idea about how many molecules of paclitaxel? Because paclitaxel is a drug with a high molecular weight. So how many molecules are linked for macromolecular pullulan?
In terms of molar percentage, it would be like something like 0.9% molar. Yeah.
So
yeah, I didn't write it down.
Do you think that you can improve this amount? Because one of the problems of macromolecular prodrug is the quantity of drug that you can link. I know that of course solubility change, improving amount of paclitaxel decreases water solubility, but maybe this amount is a little bit
little.
No. Yes, yeah.
No, that's true. I think, well, now we're at a point where we need to work on solubility, maybe first with what we have, and maybe if we are able to understand, well, There is something about pullulan, because pullulan is quite polydispersed as a natural polymer. And if we're able to get high molecular weights with a low polydispersity, we might be able actually to carry more paclitaxel. But we need first to look at this, and then also the solubility. Yeah.
Other question? Okay.
Thank you. If I understand well, you said that the aminobenzyl alcohol is a self-immolative molecule that allows you to the drug release through hydrolysis, I think. So do you have an idea about which is the rate controlling the release? Is the self-immolative of benzyl alcohol or the peptide spacer release?
Well, okay. We didn't do tests yet, but in literature it has been used and reported as cascade type of degradation. So it would be first the self-immolative spacer, this one, and then the tetrapeptide. So first
maybe first the peptide cleavage and then so you basically don't need the peptide.
Yeah, but because the interesting part is that the peptide gets away completely from the... yes.
Maybe it's faster, faster the peptide.
I need to check in more details.
Okay.
Yeah.
Okay.
No. Excuse me. My question is this one. You show that your macromolecular prodrug forms maybe a self-assembling system. But how do you think to demonstrate this self-assembling ability of this prodrug? This is the first question. The second one is how do you determine the amount of drug linked to the polymer?
The first question, I think some imaging techniques such as TEM might be an idea to confirm. About the second question, the amount of alendronate uses spectrophotometric technique by complexation of the alendronate of the polymer with ion 3. It makes a complex which is visible at 300 nanometers. So this is to see alendronate. To quantify paclitaxel, it's during dialysis. I verified the quantity of non-reacted product in the water of dialysis, with a known amount of water, of course, and a small amount of water of dialysis. So by subtraction.
But if you demonstrate the self-assembling ability, you can also think to incorporate the drug physically as a micelles.
Yes. Why not?
I have a comment. You used alendronate as a targeting agent, but bisphosphonates are also known as anti-proliferative agent in vitro, for example. So maybe that your targeting agent is also an active agent. So maybe you have any synergism between with the drug and the targeting agent. What do you think about this? Is it possible? Why not?
It's a question. No. It's a question. I don't know. It's maybe we need to do some tests to be able to say. So, but it's something which, well, it could be possible, but only tests can.
Because the indication, therapeutic indication is anti-resorption agent and so on, but a lot of papers demonstrate that these are anti-proliferative agents. So maybe that is not only.
It's true.
Targeting agent. It's also targeting agent, but not only.
Yeah.
Well, a couple of comments. Congratulations. This is a very complex project indeed.
Thank you.
Okay, personally, I like drug polymer conjugates. In the past, I used to work with Ruth Duncan, who is well known in this field, of course. But Okay, just my comments are like that. Okay, per se, a drug polymer conjugate is a new chemical entity. So here you have two drugs which are linked to the polymer. Also, I think that pullulan is not has not precedence as an injectable route, and also I am thinking of the cost of the synthesis. So either you are able to convert paclitaxel into a super anti-tumor drug, or maybe, okay, this is very exciting, but
It's true.
sorry, I'm very open, and it's really interesting.
No, it's true. We're using materials which are extremely expensive anyway from the beginning, but also the peptide synthesis is quite expensive. So yes, it has to be a fantastic polymer therapeutics to be, yes, sure.
Okay. Maybe the last question. Okay.
Thanks for your presentation. And so you showed that probably with the dimension size you can get just self-assembled system by using your product. Question is, in self-assembled form, the alendronate product, how did you imagine just the orientation of alendronate? Do you think that you can find alendronate inside the aggregates, or outside? And what could be just the potential distribution of the targeting part of your product in this form?
Well, first we need to be sure about what kind of self-assembly we have to be able to say if we have it inside or out. After, it depends. Well, it should be something, I would think it would be more outside. But I think, yeah, test needs to be done to be able to confirm these things. Those are just speculations. So, yeah, I don't know.
Okay.
Okay. So, the next.
Thank you very much.
Thank you, Dr. Bonzi.